Welcome to Ask and Learn.
Hello student, here the question is given and the question is about a person and the condition of the person is given, and here the related multiple questions are given. So the first question is about the antigen, and that antigen that cause poison IV induce contact dermatitis. So we have to tell the name of these antigen. What is the antigen? So if we answer the first question, so the antigen is going to be this urushiol. This one is an oily blend of organic substances, and these particularly having the allergic quality that is present in case of a plant, and the plant specifically belongs to these Anacardiaceae family. Anacardiaceae family plants having these type of oily blend of organic substances with this allergic quality. So the particularly the species name Toxicodendron species. So is an oily blend of organic substance having these allergic quality is the one which is present in this plant particular, and this going to cause this poison IV induced contact dermatitis. So the ultimately going to appear the rash, and leaf, stem and root of the poison ivy contain these viscous substances. So if we are talking about the part which contain these, so the leaf, stem as well as the root part of this containing these viscous substances. And the second part of the question is about the causes of these rashes and blistering skin lesion. In this case, what could be the causes? So here we can say the T cell mediated immune response. And we know this is also known as the delayed hypersensitivity or type 4 hypersensitivity. So it is a cause of poison ivy dermatitis, and in this case the red raised blistering skin lesion in that particular case. So the body immune system perceive the combination of these urushiol derivative with a skin protein as a foreign antigen, and it is responsible for this hypersensitivity, and we know this one is going to be the excessive immunological reaction that damage the body. So it ultimately going to damage the body. So this result in the skin lesion or this blistered skin. And the third part of the question is about this, the answer should be this pentadecacatechol. So it can be transferred from the initial point of contact, to other areas of the skin by the fingernail or by fingernails after scratching the itchy lesion at the primary site of hapten introduction. So it can transfer by this initial point of the contact to other area of the skin, and especially this by this fingernail after scratching this itchy lesion. So it could be the transfer. So this is why it is essential to cut the fingernails short and thoroughly wash off the skin as well as a scalp to remove the chemical and prevent further spread. So in if we want to prevent the transmission of this, so we have to cut this fingernail cut short, along with this wash off skin and scalp to remove the chemical, so it can be prevented further transmission. And the next part of the question is about the half-life of some of the protein hyphenated by the pentadecacatechol, and this can be quite long. So this CD4 memory T cell will continue to be activated. So it continue to be activated as long as hyphenated peptides are generated. As long as this hyphenated peptide generated. So the Paul case, the particular person which are mentioned in this case went beyond the 3rd week following. So this particular case went beyond the 3rd week following contact with poison ivy. Contact with this poison ivy. So here the next part of the question is how it could confirm. So here by performing a patch test. So in that particular test, the patch of this material impregnated with hapten is applied to the skin. Under seal, and this is for the 48 hours. So the area is then examined for this redness. So here the examine after this applying the area, examine for this redness, swelling or any vesicle formation. So this alternatively these peripheral blood mononuclear cells can be incubated with this hapten, or we can also do the another test. There is a peripheral blood mononuclear cell can be incubated with this hapten, and the T cell proliferation assessment. Assessed, and this assessment done for the 6 to 9 days later, because they are the immunosuppressive as well as anti-inflammatory. So in that next part of the question is this immunosuppressive and anti-inflammatory. Corticosteroids are the usual therapy for this hapten mediated contact sensitivity. So in that case, this corticosteroid is the one which is used as a usual therapy for these hapten mediated contact sensitivity. Because they inhibit the synthesis of inflammatory cytokine, because they are going to inhibit the synthesis of inflammatory cytokines. So as well as this is also going to be this arachidonic acid and its derivative, which has also performing these inflammatory responses. So we can say this PI3 prostaglandins on the thromboxane and these leukotriene. So all these which are responsible for the inflammatory responses, it can be inhibited by this. And so this corticosteroid is the used for usual therapy in that case. And how we can diagnose? So in the case of diagnosis, this presumptive diagnosis, and this can be contact sensitivity reaction to the poison ivy. And how we are going to treat it? So by applying a corticosteroid containing cream, because this is the one which is going to inhibit the synthesis of these inflammatory cytokine, and this can be used as 3 times per day. And also take diphenhydramine orally, and this is also 3 times per day. So, and another method, ask the person which are infected with this to shampoo his or her hair and wash his body thoroughly with the soap and water. Along with this, cut nail short. So by taking all these measures, we can prevent this disease. Hope you found this video useful. Thank you.